Alright, so I'm just going to record a couple of videos for you to hopefully help you understand some of the more difficult concepts over the last few weeks for this first exam if you haven't taken it already. So let's just start with a couple of questions to get you thinking about potential test questions you can see. So the first one we have here is, what would this organism be considered? And we're talking about what the way that it gains energy. So is it a consumer, autotroph, carnivore, or producer? Hopefully you know that it is a consumer. This is actually a zorse. It's a cross between a horse and a zebra. But it's a consumer. Another name for that would be heterotroph. It requires energy from something else. Remember, autotroph or producers are plants. They can create their own energy from the sunlight. So the next one, which of these is not one of the three steps of cellular respiration? Glycolysis, light reaction, Krebs cycle, electron transport chain. Hopefully this one is pretty clear that light reaction is not part of cellular respiration, but is part of photosynthesis. And then the last one, which of these describes active transportation? It requires ATP, it doesn't require any energy, moves from high concentration to low concentration. Osmosis is an example of it. Hopefully this one is also fairly clear. Active transportation means it needs energy, so it requires ATP in order to work. The rest of those describe diffusion or passive transportation. Doesn't require any energy, moves from a high concentration to low, and osmosis is an example of passive transportation. Remember, active transportation usually moves things like ions or glucose from a low concentration to a high concentration. I know some of you are having a few problems with bonding, and if we would have held a live SI session tonight, I would have gone over that a little bit more, but I'm just going to refer you back to the video I made about covalent, ionic, and hydrogen bonds before. But the big thing here is covalent bonds is all about sharing electrons, ionic bonds is stealing electrons, and hydrogen bonds is the bonding between two water molecules. Also, make sure you remember what energy set shells, how many electrons each energy shell needs. So, how many does the first energy shell need? Hopefully you remember 2 electrons to fill the first energy shell. What about the second energy shell? How many electrons are needed there? 8 is the answer. So 8 electrons for the second energy shell around the nucleus. Alright, osmosis. I know this is kind of a difficult concept, especially with hypotonic and hypertonic solutions. So hopefully this little picture will help guide us a little bit. So hypotonic solution is a let's start with hyper. Hypertonic solution is a high concentration of ions. So these little purple balls represent ions inside the water. Hyper means lots. Hypo means lack of or less. So there's less ions here, or solute as it says here. So water wants to reach equilibrium. So because there's more ions on this side and less water, and there's more water on this side and less ions, which way is the water going to want to move? So the answer is it's going to move to the right. So obviously if you looked here, it says that it's going to move that way. So it wants reach equilibrium, which just basically means there's the same concentration of water and ions on both sides. So hopefully that's kind of clear. Again, what in passive diffusion, which is what this is, or passive transportation, it moves from a high concentration to a low concentration. So over here there was a high concentration of water, and these ions can't pass through this membrane, so that otherwise the ions would also have moved. But because this is selectively permeable, only the water could move. So the water moves from this high concentration over to the low concentration, and thus we have a shift in the water due to the ions. So again, hypotonic solution is less solute or less ions, and hypertonic is lot. The way that I always remember is hyper. If you're a hyper child, then you really have a lot of energy, and so that helps me remember that there's a lot. Thinking about hyper and relating it that way. So let's see if you can answer a couple of these questions. What would happen if you put an animal cell in a hypotonic solution? So hypotonic solution would be a lack of ions. So is the water going to move out or into this cell? What would happen if you place a plant cell in a hypertonic solution? So again, hypertonic solution is a lot of ions, so less water. So what's going to happen to the water inside in the plant cell? And the last one, what would happen if you place an animal cell in an isotonic solution? I know we didn't talk about isotonic solutions, but we will in just a minute. Okay, so let's see if you answered them right. So if you place an animal cell inside a hypotonic solution, so here with this, I pulled this right off of your slide, so hopefully you recognize it. So hypertonic solution, meaning there's less ions outside of the cell. So because of that, there's more water outside of the cell than there is inside. So water is going to move into the cell. So in this case, the animal cell would lyse or burst open because the water is rushing into it, and the cell membrane isn't going to be able to hold it. In plants, it's a little bit different because of the cell wall, which is indicated here. Because of the cell wall, it's called turgid pressure. In other words, that's what helps plants defy gravity and grow up. Is because all the cells swell and they push against each other, and that's how they can stand up. The next question was, what if you put a plant cell in hypertonic solution? So now there's a lot of ions on the outside, so there's going to be more water inside, higher concentration of water inside the cell than out, which means the water is going to move out of the cell. And the cell will become shriveled. Same with animal cells, it will become shriveled, water will leave. The isotonic solution is basically a solution that has the same concentration. So there's going to be the same concentration of water and ions inside the cell and outside. So water is going to come. So as water comes in, water will leave. So it just maintains the same concentration of water in and out. Alright, so now let's move on to monomers versus polymers. Sometimes this is kind of a hard concept for people to understand. So monomers are the building blocks. So I like to compare monomers and polymers to a chain. So the links of the chain would be the monomers. So each link is a monomer, but the chain as a whole would be a polymer. And monomers and polymers come into play when we talk about the molecules of life, like proteins, nucleic acids. So the monomers of a protein is amino acids, where the polymer is the protein itself. and the monomer of nucleic acids would be nucleotides. So hopefully that helps a little bit. So again, building blocks is the monomer, so each little link. So this would be, if this was a protein, this link would be an amino acid. This could be a different amino acid, another amino acid. And all of those make up the polymer or the protein. Now there's a couple of questions about saturated versus unsaturated fats as well. Also a concept that can sometimes be hard to grasp. So here's a couple of diagrams. So saturated fats is all about being saturated with hydrogens. So here we have a chain of carbons right here in the middle, and each carbon has a hydrogen that is linked to it everywhere that is possible. So you can say that saturated fats are completely saturated with hydrogen atoms. Everywhere a hydrogen atom can be, it is. On the other hand, unsaturated fatty acids have less than the maximum possible of hydrogens, and they have double bonds, which is indicated right here. And these double bonds create a little kink in the molecule. So this is just another depiction. This is a unsaturated fatty acid. Notice the big kink in it. This would be a saturated fatty acid, and this is trans, but we don't need to worry about that right now. So unsaturated fatty acids, they are liquid at room temperature, and it's all because of this kink right here. If you think, yeah, this is saturated, which are solid at room temperature. If you think about it, these saturated fatty acids, they can stack really close to each other because they're flat. And because of that, it makes them solid. But these unsaturated fatty acids, because of this double bond putting a kink in it, they can't stack as close. And so they wouldn't be able to be as close to each other as in saturated, and that's what makes them liquid at room temperature. So plant oils or whatever else would be the unsaturated fatty acids. So hopefully that helps with these few concepts. If you have any questions, feel free to email me if you are having trouble with any of these concepts and you haven't taken the test yet.